Hello, my name is AmandaKowalczyk, and I'm so grateful to be here today to present my research to all of you. Thanks to the organizers for putting this symposium together, as well as to you for coming to listen to my talk. I'm currently a postdoctoral fellow with Nathan Clark and Maria Cecchina at the University of Pittsburgh, but I just defended my dissertation in May, so I'm going to talk to you about some of my dissertation research that involves identifying the genomics underlying extended lifespan in mammals from a comparative genomics perspective. We want to be able to better understand the genetics underlying longevity and aging to better serve human health concerns. We want to understand why humans age, potentially delay aging, extend lifespan and health span, as well as tackle age-related diseases. Unfortunately, whenever we're studying the genetics of longevity or aging, it becomes a very difficult problem because these are such complex phenotypes that there are probably many genetic elements at play driving those phenotypes. So it's difficult to directly connect whatever's going on at the DNA sequence level to the phenotypes that we observe. Mammals are a really great system to try to tackle this problem because they have a really enormous range of potential lifespans, from hundreds of years in whales to just a couple of years in small mammals. So by using that really huge phenotypic variation, we were hoping to be able to get a better perspective of what's going on at the genetic level to lead to those really diverse phenotypes. The way that we do this is by using convergent evolution, which is the process by which unrelated species develop similar characteristics. The sort of Bio 101 example of convergent evolution is the flight phenotype, in which birds, bats, and the now extinct pterodactyl independently acquired the ability to fly. When we see this type of convergence at the phenotypic level, we may also expect to see convergence in genomic elements associated with the phenotype. So by looking for concordant patterns of convergent evolution in genotypes and phenotypes, we can connect those two pieces of information and find genotypes associated with our convergent phenotype. We developed a method in the Clark and Cecchina labs to perform this exact task, to link convergently evolving genomic elements to convergent phenotypes. It's called RER Converge. It's a really great method that can run very quickly on genome scale data. It can run just a couple of minutes. And it's very flexible to any kind of input you want to use, any kind of genome sequences that you have aligned from any set of elements, genes or regulatory elements or whatever. It also can take any kind of convergent phenotype that you want to study. So just as a little plug for RER Converge, it's available on GitHub to download for R if you want to try it out on your own genomic sequences and your own phenotype of interest. The way that RER Converge works is that it starts out with something called relative evolutionary rates, or RERs. These start out as just plain old evolutionary rates that are calculated as branch lengths that represent the number of substitutions that occurred between a species and its ancestor. They're converted from evolutionary rates to relative evolutionary rates through some statistical corrections as well as an average evolutionary rate normalization. So each branch is normalized for the average rate of evolution genome-wide for that particular species. We have one of these trees for every single one of our genomic elements, so that could be 20,000 or hundreds of thousands of genomic elements depending on what you're looking at. And in tandem with these RERs, we have trait information. We know what our phenotype values are for our living species. We can use that information to predict what they were for ancestral species, and then the branch lengths on this trait tree are just the change in phenotype between a species and its ancestor. We put these two pieces of information together, match up branches that go to the same species, and perform correlation analyses to find correlations between trait evolution and the relative evolutionary rate of genomic elements. If we see a significant correlation here, that indicates that the genomic element is potentially associated with our phenotype. And I'll point out again that we do one of these statistical tests for every single one of our genomic elements. So there would be 20,000 or hundreds of thousands of statistical tests run to test for these correlations. So to be able to use that method to find genomic elements associated with longevity, longevity must be a convergently evolving phenotype. To see if that's true, we can look at our phylogenetic tree of 61 mammal species whose genomes are available through the UCSC 100-way alignment, as well as maximum longevity values for those species that I got from the ANAGE database. I can pull out very long-lived species, and you can see that they're very distant on the tree. They're distantly related, as well as short-lived species, also distantly related. So since these species appear to have independently acquired these traits, that indicates that yes, longevity is a convergently evolving phenotype. But whenever you're studying longevity in a comparative context, there's always a caveat that lifespan is really strongly confounded with body size. Species that are long-lived also tend to be really large, and species that are short-lived also tend to be really small. We were interested in the evolution of that phenotype, evolving to be large and long-lived or small and short-lived. But we also were interested in sort of the guys that don't follow that trend, species that are able to evolve to be long-lived without growing to be a large size. So we constructed new phenotypes to tackle those two convergent phenotypes that we were really interested in. And we did that using principal component analysis, and I'm going to step through really quickly how principal component analysis works in 2 dimensions. So if I look at a plot of my body size for my mammal species versus lifespan, I can see that there's this really nice trend between body size and lifespan. So my first principal component between these 2 values is just the line of best fit. For each point, each species, I would project that point directly down onto the line, and its distance along this line is its principal component 1 value. So each species gets a numeric PC1 value. The extreme species of PC1 are those on each end of this line, and indeed those species are things that are large and long-lived and small and short-lived. So I call my PC1 phenotype the long-lived, large-bodied phenotype. On the other hand, PC2 is exactly perpendicular to PC1. I calculate the values the same way, and my species with extreme values for PC2 are things that we love to study when we're looking at longevity, like bats and the naked mole rat that are very long-lived given their body size. And I guess hoofed mammals sort of got the short end of the stick on the other end of that spectrum. But we call the PC2 phenotype then the exceptionally long-lived phenotype, or species that are exceptionally long-lived given their body size. So moving forward, these are the 2 longevity phenotypes that I plugged into RER Converge to run my analyses. So I ran RER Converge with 19,149 genes from those mammal species I described earlier. These are p-value histograms both for the 3L and ELL phenotypes. In red is the real p-value distribution from using my real phenotype values, and in blue is an empirical null distribution. And what I really like to see in both of these plots is that there's a nice enrichment of red over the blue on this left side at low p-values, both for the ELL and 3L phenotypes, which is indicating that there are genes who are significantly correlated, whose rates of evolution are significantly correlated with my phenotypes, even after correction for multiple hypothesis testing. So this is indicating that yes, there's real statistical signal associated with my 2 phenotypes. After doing this at the gene level, I then proceeded to perform pathway enrichment analyses to see if there were whole groups of genes who were evolving in concordance with my 2 phenotypes, and those are the results I'm going to show you. So looking at the evolution of the 3L, or the long-lived, large-bodied phenotype, these are my pathway enrichment results, where each point is a pathway. The shape and size represents how enriched that pathway was, and the lines between the pathways represent how many genes those pathways have in common. And by defining the number of genes in common, I was able to make these clusters to correct for pathways that are very, very redundant and look at large functions associated with the 3L phenotype. Now I'll point out that regardless of the size and coloration of these points, all of these pathways were significantly enriched in association with 3L. So what I see in these clusters is lots of cell cycle pathways, lots of cell death pathways, lots and lots of immune pathways, as well as these Fanconi anemia pathways that are DNA repair pathways associated with the long-lived, large-bodied phenotype. And finally, I'll draw your attention down here to these singleton pathways that just didn't have a ton in common with the other pathways in these clusters, so they didn't cluster. But there's lots of longevity buzzwords down here in these singletons, like the IGF-1 pathway, which is associated with lifespan in C. elegans, telomerase pathway, that's telomeres, are a really important area of study for lifespan, as well as p53 regulation, and p53 is a essential DNA or cancer control gene, and that's a really great segue into what I want to talk about next, which is why I think all of these pathways are associated with the evolution of the 3L phenotype, and that is that they are all associated with cancer control. So if we think about how cancer happens, it starts out as DNA damage that results in unchecked cell cycle progression and or inadequate cell death. Those cancerous cells then evade the immune system, and in some cases inflammation even promotes tumor growth. And then later stages of carcinogenesis and tumorigenesis progress. So what I see is pathways associated with all of these early stages of cancer control evolving in association with the long-lived large-bodied phenotype, and that helps to resolve something known as Pitos Paradox. Pitos Paradox is the observation that large species should get cancer more often than small species just because they have more cells, but we don't actually observe that. So if we look at a plot of body size versus the rate of cancer in species, we would expect this blue line where large species get cancer more often. Sort of your probability of cancer increases as you get larger, but what we actually observe is about constant rate of cancer across species of all sizes. And here's a paper that shows one possible explanation for that, which is this is body size versus mutation rate. So as you get larger, your mutation rate actually decreases, which helps to resolve this paradox. But my work finds that it's potentially tons of different cancer control mechanisms that are working at higher efficiency in long-lived, large-bodied species to allow them to evolve to those states. And this is something that's really universal across mammals and not just unique to specific species. So that's the 3L phenotype. If we look really quickly at what's going on with the ELL phenotype, or species that are exceptionally long-lived given their body size, I found some really cool results. This is the one that I think is the most interesting, and that is that DNA repair is uniquely associated with the evolution of the ELL phenotype. Each of these plots is showing enrichment for a different DNA repair pathway, and in orange are the results in association with the ELL phenotype. So in all of these plots, what we want to see is this nice uptick. On the right end, this is showing enrichment associated with the ELL phenotype. And we don't see the same trend if we look at the gray line, which is the 3L phenotype. In some cases it's sort of there, but it's much weaker and not significant. So this indicates that this is really unique to the ELL phenotype, which is in support of some work that suggests that DNA repair and efficient function of DNA repair mechanisms is really essential to extension of lifespan independent of increased body size. So really cool stuff here. So in conclusion, one really great result from this work is that we could actually perform these types of studies using continuous phenotypes, using really, really large data. RER Converge scales up to like hundreds of thousands of regions and hundreds of species. So it's a really great method as we move into the era of having lots and lots of genomes sequenced. And in terms of work that allows accessibility of science for all, I really love this research that I've been able to contribute to because it all uses existing data that's freely available software, so anyone, anywhere can do this kind of work. And the results that I found, of course, is this potential resolution to Pitos Paradox, that efficient cancer control is essential to evolve extended lifespan universally across mammals, as well as potentially DNA repair associated with lifespan extension independent of body size. So thank you to everyone in my labs who's contributed to this, as well as collaborators. I wouldn't have been able to do it without all of your great support. And thank you all again for listening. Please feel free to reach out to me via email or Twitter if you want to network or chat more about this work.